So today, I'm going to talk about our core facility at Columbia University, and I'm going to tell you about kind of like our initiatives and our experience from two perspectives. One is my own as the director of operations, and the other one will be from a very talented junior team member, Chris Wu, is going to tell you about his experience in the core facility. So, let's see. There we go. So our affiliation at Columbia is with the Columbia Stem Cell Initiative, which is really Columbia's interdisciplinary stem cell community. So it's comprised of about 70 investigators whose common interest is in regenerative medicine and stem cells. And our director, Emmanuel Passague, has really spearheaded this initiative. She was recruited in 2017 to Columbia, and I actually started with Emmanuel back then, and she's really built up this program to an amazing entity at Columbia University. So some of the research focuses of CSCI, of course, the overarching focus is regenerative medicine and stem cells, but among these 70 investigators, there's a focus on development, there's a focus on neurogenesis and neurodegeneration and treating diseases of that nature, disease modeling, and especially CRISPR-Cas9 editing of patient-specific tissues, tissue engineering, blood, and also aging studies. So even though we're part of the Columbia Stem Cell Initiative, our user base in the core facility is institution-wide. So we have users from all three Columbia campuses from a variety of different research interests and departments. So our facility, everything we do is kind of guided by a philosophy we've developed, and really it's centered around providing a model that is flexible and adaptable to users, flexible and adaptable access to well-maintained instrumentation, and through this model to enable users through training, SOPs, and other educational materials and support. So really, this flexible and adaptable model is facilitated through a lot of support and training and materials that we've developed. So as part of that, we have a very strong emphasis on independent access to the instrumentation, including the cell sorters. So we do a lot of training in the facility. And as I said, we've developed a lot of materials, including SOPs and other educational endeavors. And we have about over 80% of the cell sorter usage is independent by researchers, and way over 90 of cell analyzer usage is independent by researchers. And the choice of instrumentation that we brought into the facility has been heavily influenced by this model. So we have about 550 active users, 200 PIs, about 200 labs across campus. And as I mentioned, we have campus-wide representation. So the labs are from both the medical center, the Morningside campus, and also the Neuroscience Institute, JLG Center. And we're very fortunate to have a centralized location in the same building, and we have 3 rooms. We have a cell sorting room, our cell sorters, we have an analyzer and imaging cytometry room, and then we have a genomics room for our 10x genomics. I'll talk about that in more detail in a little bit. And last but not least, definitely not least, is the team that we have established in the facility. So I am very fortunate to work with 3 very talented and enthusiastic team members, Juan, Ryan, and Chris. And I think enthusiasm for me is the most important trait for working in a core facility, because it is not an easy job a lot of the time. And being enthusiastic and excited about work goes a long way in this kind of role. So I'm very happy where we are with our team. In terms of our instrumentation, so we have 4 conventional cell sorters. So we have 2 Sony MA900s, 2 Arias, and then our conventional analyzer platform is solely NovaCyte. So all of our conventional analyzers are NovaCytes. We brought that technology on a few years ago. We were also very fortunate to receive our first NIH S10 grant for the image stream several years ago, and that's become a very important tool in the facility. And then we recently got our second S10 grant for the FACSDiscover, which was just installed a few days ago. We also have Spectral Analyzer is the ID7000 platform, which we hope to bring more of those instruments online in the future. And we also have access to genomics. So we had the Chromium controller for a few years, and we just got the Rhapsody platform. So we're excited to get that online. And we also maintain other kind of complementary instrumentation like cell counting. We have qPCR machines that people use and some cell preparation instruments like the Miltenyi GentleMax. So working in a core facility requires a very unique blend of skills of multitasking, of course, the technical and scientific aspect, and then there's this customer service aspect. So it's a very unusual type of role because it's a mix of really technical stuff and customer service. So it's becomes definitely is an interesting experience a lot of the time. But in terms of I'll talk more about that in later slides. But in terms of our activities, just what we spend our days doing. So we do a lot of operations management. So that entails scheduling, user onboarding, other administrative work. We also do all our own budgeting and finance and purchasing in the facility. So a lot of that's on us. As I mentioned, strong emphasis on user training and education. So a lot of time spent hands-on training, and also a lot of our time is spent making documents and SOPs and educational materials for users. I actually personally really like doing that. So it's definitely an enjoyable part of the job for me, but we do spend a lot of time on that. Of course, instrument maintenance, goes without saying. We have some assisted usage, so we do spend time operating instruments for users. But we do spend a lot of time on experimental design, data analysis support. We help people design panels, we help people troubleshoot experiments, design experiments. And because the research being done at Columbia is very diverse, there's neuroscience, there's immunology, there's biomedical engineering, tissue engineering. So it's really, every day it's something different. And when we help people with this experimental design, it's very often an entirely different system that we're working with from day to day, different animal models as well. And then finally, we do a lot of kind of ad hoc troubleshooting and technical support. So we're kind of on call throughout the day where there's always someone present in the lab space. People know how to reach us. So we're constantly helping people with issues that crop up on the instruments. So to give you a little bit of history of the facility. So I started in 2017 with Emmanuel Passaguet, who's the director of CSCI, and we purchased 2 Faxaris and a Celesta, so we had 3 instruments. And then I was very fortunate to get our first staff member, first team members. The first time I managed somebody, Dan Trost, who started in 2018, and he's gone on to develop a very rewarding career in cytometry, and I'm very proud of him, what he's accomplished. So he works in R&D and instrument development now. And then we got our first S10 grant for the image stream in 2019. That was another big accomplishment. And this is a very important instrument for our facility. It's used a lot, and we constantly are developing new applications for it. We're working now we're really getting to EV work on it, which has been very interesting, a learning experience for us. And then a second very special team member, Rose Gordon, also joined in 2020. So she's also gone on to develop her career in cytometry in another context. So I'm very proud to have 2 very excellent people that were trained in our lab that are working in the field now. So, and then in around 2021, we got our first NovaCyte, which I said has become the standard conventional analyzer platform in the facility. So when we got this instrument, we didn't really know much about it. It was kind of a new, not very common in core facilities, but we were so happy with it that our 3, the 3 conventional analyzers in the facility are NovaCytes now. We purchased 2 after that. And then big accomplishment was getting the Sony platform in 2021. So this really expanded our ability to train users on the sorter. And we did that with the ARIAs, but it's a lot of work. It was not that effective. We still do it, but the ARIA is not as easy as everyone probably knows to train people on. So when we got the MA900 platform, it really opened up a lot of opportunity to get people trained on the sorters. And then another very interesting thing that we also endeavor that we also undertook in 2021, 2022 is bringing on a self-service single-cell genomics service into the facility. So we got a Chromium controller and we rolled it out as a instrument that we train people on for after-hours access for 10x genomics, which I'll talk a little bit more about that as well on a different slide. And then we got our first spectral instrument last year, and finally we got our S10 for the FACS Discover, which I mentioned. And now where we are now is we have, five sorters now, 4 analyzers, image stream, and a great team. So I'm very proud of what we've accomplished over the last 7 years. So just to highlight some of our activities and facilities. So I want to talk about our program for independent access to cell sorting, and then also talk about the genomics technology. So with our policies and our program for independent sorting, so trained users can access the instruments on essentially unrestricted basis 24/7. So this is really important because people have very unpredictable clinical samples, for example, and it just allows the throughput to be so much more because the sorter access is now restricted to staffed hours. So we have people often sorting to close to midnight. The sorters are running from 9 a.m. like well past 6 p.m., sometimes much later in the evening. And in order to do this, we knew we had to develop a very comprehensive and standardized training program for this, including documentation, SOPs, etc. So we spent a lot of time developing that for both of the cell sorter platforms. And as I mentioned, we have over 80% of the cell sorting usage is independent. We have almost 300 users trained on cell sorters, almost 200 on the Sony, and then over the years, we've trained about 80 people on the Aria. So to highlight our Sony training program, so we have, it's mainly comprised of a 3-hour hands-on session, and then we also have ad hoc support as necessary afterwards. So the kind of formalized training is a 3-hour session, hands-on session. And one of the things we developed is an SOP for us for the training. It's not only an SOP for the users, but we have an SOP of how the training should be done. So every team member that does the training will be able to train users in exactly the same way. So that's worked out really well. And we also found that it's very effective for us to provide samples. So during this first session, we provide samples that we prepare for the users. We discourage the users from bringing their own samples. The argument, of course, can be made that everyone's experiment is different and they need to be trained for their particular application, but we help them with that during follow-up sessions. But we really like that the samples that we provide are able to facilitate training users in every aspect of instrument operation that they need to know, with compensation, without compensation, 4-way sorting, etc. And then we also have our printed material. So we have a guide that they get. It's about 50 pages that we put together for the Sony. So it walks them through the setup and setting up an experiment and everything they need to know, troubleshooting as well when running the instrument. And then they also get printed SOP, and also we have a troubleshooting guide. And all these materials are available online as well. So I also want to emphasize that we're open to sharing any of the materials that we've developed. So I should have put a QR code on the presentation for that, but we don't currently. We're changing the way we're managing the SOPs now, so that wouldn't have been that effective. But please reach out to me, and we're willing to share SOPs for any of the instruments that we've developed. And then we'd cover kind of just the basics for the Sony training. So we teach them how to set up the machine and run auto calibration, setting up an experiment, multi-way sorting, cleaning and shutdown. So I mean, we have users come on the weekend often, like they don't even tell me, and they send them on the weekend and there are no issues. I mean, no one calls me for issues on these instruments. So it's worked out very well. It's really helped a lot of people accommodate difficult experiments, and we're excited to see what new instruments we'll be able to bring online with similar workflows that facilitate this independent access. And we're in the process. Our S8 will also be accessible to users independently, and we're working on our training program for that now. It's going to be more complicated, but we definitely will roll that out as an independent instrument. So for genomics, so we decided to integrate the 10x Chromium controller in the facility to fulfill an unmet need, which is the availability of after-hours access to the next-gen workflow. So this would allow users to take cells that were maybe from an unpredictable model or clinical samples and be able to make stable barcoded cDNA that can be stored for single cell sequencing after hours, because the current services at Columbia in the Genome Center, they have a cutoff time of around 5 p.m. So this would allow users to be able to utilize the technology after hours to accommodate these difficult or unpredictable samples. So the way we decided to integrate this is through a collaborative model with the Genome Center. So of course, Columbia has an excellent genomics facility. It's the single cell core in the Columbia Genome Center. And of course, we did not want to duplicate their efforts because they have a full service 10x genomics workflow. So we spoke with them and we said, we're going to integrate this 10x Chromium control into the facility. And we worked out a kind of a collaboration where users can obtain 10x reagents from the single cell core, so they don't have to buy reagents from 10x. They can use our platform, and we provide training for this. So the genomics facility vetted our training and reviewed it. And then the barcoded cDNA that's generated by the users can be processed by the Genome Center through their workflow of library prep and sequencing. So really, it worked out really well. It's very collaborative. It fostered a lot of communication between the flow cytometry core and the Genome Center. And it's in addition to working out well just as a service, it's gotten us much closer and collaborative since we worked this out together. So why this works, it avoided that redundancy on campus, because definitely with cores, we don't want redundancy. And then, as I said, it fostered this collaboration. And the other thing is it really tapped into the existing knowledge at Columbia. For me to, at that point, integrate the Chromium and train people not only on the gem capture, but also the library prep and the sequencing isn't the best use of the skills, because I don't have a lot of molecular biology skills. We work with whole cells. The Genome Center is an expert on that. So it worked out really, really nicely to take advantage of the expertise in each respective core facility. And we just got the Rhapsody, so the BD single cell analysis platform. We're going to also roll that out as a self-service instrument, and we're excited to explore that. So before I hand it over to Chris, just to talk about things that I really like about my job and what's very rewarding, is because it's not always the easiest job, but there are things that are very rewarding for me. So for me, just being able to be exposed to such a diverse array of research areas, because as I mentioned, Columbia University has many, has many different departments with many different focuses, neuroscience, immunology, cancer biology, biomedical engineering, et cetera. So I'm really able to support research from many, many different types of research interests. And I'm constantly learning. I mean, for me, I actually don't have a degree in biology. So this is like, I feel very privileged to be able to support this kind of research, to work with such smart people at Columbia. And I also learn a lot all the time. Sometimes I wish it was a little bit more boring, but I really learn something new every day. And I said, the ability to contribute to this cutting edge research, because I don't have like a very traditional science background. So it's never boring. It's always a challenging environment. And I also am very fortunate, and I really enjoy mentoring team members for future careers in cytometry, which has been, as I mentioned with Rose and Dan, it's been really nice to see them develop their career. So in terms of challenges, I think for me right now, it's really about growing pains, because we've grown from a facility with 3 instruments and just like a handful of users now to over 500 users. And we have not prepared for this type of growth. So I think for me, the biggest thing that bothers me is the limitation on the amount of attention that we can give each user. So I really enjoyed, like 4 years ago, when I was able to work with each researcher much more closely to develop their project and develop their experiments. Now I just don't have the time to do that. I really don't like not being able to give each researcher my full attention. There's no way to do it right now. The volume is just too high, but that is like not the best thing. And we're trying to work out plans to be able to support researchers more effectively, but it's been a pain. The other thing is our operational system. So what we used for scheduling and coordinating communication with users really has not kept up with the growth of usage. So we are now developing much more efficient systems to handle this like insane volume of usage. And that's something we're working on now to develop. And the other thing which also drives me crazy, which is tied into the first point about attention to users, is I just cannot oversee the data they're generating as much as I used to. So I really don't know a lot of times what they're doing. I can't review every FCS file they're generating. I can't look over everyone's shoulder. So I'm terrified that people are not doing things the right way and generating not great data. So this is something that's, I think everyone kind of encounters this problem in a high volume facility, but it's something that definitely keeps me up at night for sure. The other thing is more of the customer service aspects, is just managing expectations from researchers and also many different types of personalities. So this is, you have to have a lot of patience to do this job. And sometimes I don't have that much patience. I find myself having less and less patience, but this is that customer service aspect, which is, as the volume increases, this also becomes more difficult because there are more people, just the numbers of people with unique personalities. And then with a high volume facility, the need to multitask really becomes very unproductive. So it's just when things are too hectic, it's just too chaotic. I mean, it's really like dealing with the chaos can be very unproductive and frustrating. So we're hoping that the new operational systems that we're developing will help with that chaos. But that's all I have to say. I really want to have Chris speak. So Chris is a very kind of, I think, unique team member. He reached out to me a year ago because he wanted to work in a core facility. He had just graduated essentially from college, and he wanted to work in a flow cytometry. He never really had much experience with a flow cytometry core. So he knew what he wanted to do, and he was very proactive about trying to find a job in a core, and he really has grown immensely. He's become an essential member of the team. He's extremely talented, and he has kind of some interesting things to say about his experience working with us.
Hello, my name is Chris. I am a very fortunate member at the CSEA Full Corps. So today's talk is going to be kind of like a career development talk, to be very relaxed, very, informal. So I kind of framed this talk by just kind of introducing why and how I actually joined a academic flow cytometry core. And then the second part of my talk is to be like a quick 10-minute talk, but I'm just going to talk about some frameworks that kind of help me navigate a academic core facility. So to start, a little bit about my background. So I was your traditional pre-med undergrad chemistry major back in undergrad. So once I graduated, actually about like a year and a half, 2 years ago, I, kind of decided medicine really wasn't for me. So I was just kind of looking around, trying to find, what was like kind of my calling. So luckily enough, I feel like we're very fortunate as the flow cytometry community to have a lot of scientific communicators. So there's a plethora of offline resources in addition to like podcasts, different like YouTube videos, and then also like community spaces where people just talk about flow and then people actually teach flow. A couple of these resources that I did want to mention was there's like two particular podcasts that I'm not sure if people have heard of, but Flow Stars is one. It's produced by Beckman Coulter. Just talks about some of like the bigger names in flow cytometry. Also, luckily enough, I think Ruhi over here, he was a part of something called Open Flow Cytometry on YouTube. So I think during COVID, Ruhi and his group of people, so I think it was Kathy Daniels and then also Derek Davies, they decided to put up a bunch of like tutorials about how do we do sorting, how do we actually do flow analysis, data analysis, and everything related to flow cytometry. So with both those resources, so like podcasts, YouTube, and then also, OMIPs, and like the very conventional Practical Flow by Howard Shapiro, I actually reached out to Rui, and Rui actually like referred me to Mike, and here I am now, speaking to the MetroFlow community. So coming into the core facilities, I had some expectations of, what a core facility was going to be. So just to preface a little bit, so my undergrad was a predominantly undergraduate institution. So we had no postdocs, no grad programs. So the idea of having like a core facility, zero, not. So everything, that comes from the core facility, I mostly picked up from like podcasts and then other offline materials. So when it came to working at a core facility, the first thing that kind of like really drew me in or kind of gave me a why was kind of like the ability to develop technical expertise in a specific topic. So, like coming into a academic core facility, I was like thinking, right, like if I was working at an academic core facility, I'll be like some type of like wizard of flow cytometry, right? Being able to do like experiment design from the beginning, doing like panel design, operating instruments, and then doing all these like different functional assays afterwards, or even like characterizations via sequencing, etc. In addition to that, something that really drew me into flow cytometry was the community aspect and also the collaboration aspect of flow cytometry. So because this is a core facility, right, we serve a community of different PIs, different users, and we are supposed to be like the de facto technical experts of flow cytometry. So like in my very naive thinking, right, I was thinking, technical expertise, that must mean like domain expertise, right? And then once I get that, publications, co-authorships, easy, right? Everything's going to be like kind of coming to my feet, super simple. And then last but not least, one thing that really drew me into the core facility aspect too was the prospect of doing our technical development. So in my previous research of flow cytometry, I saw like different like interesting techniques, right? So there's this one sorting technique called like LibreC, stands for linking amino B cell receptor antigen. So essentially all this does is you conjugate a like PE antigen to or a PE fluorophore to an antigen, and then from there you're able to essentially sort out any like PE positive like B cells and then sequence it afterwards in order to find like the BCR sequence of that like particular antigen. And like the prospects of like being able to do that at like an academic core, I was like, oh yeah, that's crazy, right? Like I would love to do that. Well, reality, very different. So when it actually comes to, working at a core facility, I feel like the realities are much, much different. So first of all, when it comes to flow cytometry, right, you're not supposed to be an expert at everything. So when it comes to like in vivo techniques, for instance, I've never scrubbed a mouse before. When users come up and ask me like, oh yeah, do you have any like tips for scrubbing a mouse? Zero, not. Absolutely no idea how to do that. If you ask me any like flow questions, oh yeah, like what fluorophore should I add? What marker should I like kind of use? I can maybe help with that to a certain degree, but anything like experimental design, if you want to like cross a mouse, do anything like that, you gotta ask like an immunologist. So yeah, like working at a core facility kind of like honed down my like expectations of what I can actually do at a core facility. So in addition to that too, right? Because I'm not an expert at everything, right? I'm not this like flow wizard or experiment wizard that can do everything in science. Now, co-authorships and maybe in the future when I know a little bit more, but currently, just mostly focusing on doing my best to provide good technical expertise when it comes to doing flow cytometry. And then last but not least, tech development, right? If we're not doing any co-authorships, if we're not like experiment experts, then the prospects of doing, tech development kind of decreased. But one thing that actually I do want to bring on the positive side is when it comes to working at a core facility, I feel like you do get a lot of autonomy when it comes to operating your instruments. So one benefit of working in a core facility environment is the prospects of doing operational development, still development, right? So one thing like that we've been, kind of rolling out and trying to do is introducing like new flow techniques to our users, for instance, using an image stream to do extracellular or EV flow cytometry. In addition to that too, since we have a little bit more control about the instruments themselves, I've taken some liberties of kind of like upgrading our computers, for instance, from hard disks to SSDs, and then also like implementing some like new operational changes. Like for instance, we have a automated manner of doing flow cell cleans and ARIAs versus having to manually like click cytometer, cleaning settings, and then flow cell clean. So now for some frameworks that I feel like has helped me, really adapt to the core facility life. So the first core or like the first framework that I feel like has helped me a lot is going to be like realizing that domain knowledge is key. And sadly, we don't have that domain knowledge. So when it comes to really succeeding at a core facility, we really have to work with our users to essentially get what they want, right? So if a user comes, we have absolutely no idea what they want, sadly, we can't help them, right? We're not immunologists, we're not like geneticists, right? If they want to sort something specifically, we can definitely help that, right? We can help them find markers. Maybe I'll do a little caveat there, but we can help them at least like determine like which fluorophores would work best for their panel. In addition to understanding our limitations, it's also really important to keep in mind that working at an academic core facility is really a service job. So because this is a service job, we have responsibilities to our users, right? We should be, communicating with them like relatively well. If there's any issues with the instruments or if there's something that's like outside of our control, just let them know, right? You can't communicate if you don't know what exactly is going on. Lastly, one of the core tenets that I did want to point out was that all core facilities are not the same, and then same thing with users, right? Not all users are the same. So it's very important to stay flexible, but also understanding like what the needs of the users are to really help them with their experiments. So putting this all together, right? The core facility life, very difficult, right? It could be very chaotic. It's really hard to standardize, this user from another user. It's very difficult to really take in all that information, right? We have like users who are doing like iPSC differentiations, and then we have some users, right, that are just doing straight immunology. And then it's very difficult to really like understand everything to an exact detail. So it's really important to be able to kind of take it to a certain point and then be like, user, you can tell me what I need to do, but then from there, I'm going to be like that technical expertise, or I'm going to give you that technical expertise to execute what you want to do, right? Whether this be like single particle analysis, single cell analysis, single cell sorting, etc. And yeah, so I just want to make a few acknowledgements really quickly. So I want to thank the CSCI Flow team. So Ryan, sadly, he's not here because he's actually sick, or he's been sick for the past couple of days. We have Juan. I would also like to thank our community and then also our like the CSCI Flow users. Because without them, right, we wouldn't have a job, we wouldn't be helping anyone. So we really appreciate that. And I also like to thank Emmanuel Pasike. She's the head of our CSCI community, and she's really actually done a lot to really bring this community together. Last but not least, I really cannot forget our field support engineers. Shout out to Bola, John, Kaushal. I know John's definitely helped us a lot. We've been having issues with our ARIAs, but not to forget, Anthony, Tim, Carla from the Sony team, and then also Alex from Agilent. But yeah, thank you. So, thank you guys for the section.
No one has any questions for either Chris or Mike?
Is this your first talk, Chris?
It is my first talk.
Good job.
Appreciate it.
Like why flow cytometry?
Great question. So I kind of like skipped over this in my background, but I actually had a little bit of flow cytometry experience in my undergrad institution. So I did some undergrad research doing single particle analysis on lipid nanoparticles. Albeit now that I know a lot or a little bit more about EV flow, I feel like some of the work we did was maybe kind of like Wild West out there. But at least it introduced me to the technique of flow cytometry. And then actually one thing I do want to point out, I feel like the thing that really drew me into flow cytometry was the fact that it's single cell, single particle. You cannot get that with like ELISAs, like plate readers, like PCR, single cell, single particle, flow cytometry, amazing.
It's just nice to hear someone who actually picked flow cytometry versus the rest of us who think it does. So
Great talk, Chris. Here.
Oh. Hey Rui, nice to see you.
Hello. So, this is from a perspective from academia. So how do you keep people like you from moving to industry? Because, no bias at all. Because, when I hear somebody like you talk, my first thought is, okay, I'll give him 2-3 years and he's gone. So how do you keep people like you?
So I'm gonna give you a fake answer first. It's money, but I feel like there's definitely a lot of like career advantages at working at a core facility. I feel like working in industry, right, you can be like kind of honed in a specific subsector. Like I was actually talking to like a postdoc or like an assistant professor who actually used to work in industry. And it was really cool to see how much he knew about like, lentiviral transductions, for instance. But I feel like, he comes to the core, right? He doesn't really know too much about flow cytometry. So I feel like if he, someone who like knows so much about like a specific topic still needs, some help with flow cytometry, I feel like, I've actually like kind of like in a nice place, right? Where I'm like learning from like domain experts and then also getting like a really nice community to really interface with.